from CMAAO 112th session. We meet every Saturday, 9:30 Indian Standard Time, and we meet for talking about what is going around the world. These are National Medical Association meetings, which happens every month, every week. This is a weekly session. So today we have a topic of origin of COVID-19 and the evolution of its variant, the past and the future. So to present this topic, we have Dr. Pavitra Venkatagopalan. She's a PhD in microbiology. She's a director for Care Diagnostic Center, Chennai, which is an NABH certified medical laboratory. Welcome, Dr. Venkatagopalan. And this session is about, so we are meeting for the last 2 years in this session, and we are discussing about pandemic, we're discussing about all other issues. So the platform is all yours to present.
Thank you, Mr. Agarwal. I will go ahead and start with my presentation. Okay. Good morning, good evening, good night to everybody that's joined us from all around the world here. My relationship with Dr. Agarwal's team is quite an interesting history. When Dr. Agarwal first reached out to me to talk, I was due for delivery any day. And he said, can you commit a particular date? I said, I can't, I have to wait to see if when my baby comes. And then ultimately my baby cooperated each time I had to come and give a talk with Dr. Agarwal's team. My background is that I have a PhD on coronavirus studies, and I studied at Arizona State University. I graduated in 2012. This was at a time, our university had a lab set up where we had an open lab set up. The 2 bays next to ours was working on Ebola virus, another bay was working on smallpox, and we were the coronavirologists in the middle. And we'd always joke about how, these guys are working on such scary viruses, and here we are working on a virus that simply causes the common cold. And lo and behold, 10 years later, we are sitting here talking about a pandemic that is caused by coronaviruses. Since the background of all the attendees are quite varied, I will be going through a little bit of the background to ensure that we're all on the same page. The first question that we had to explore when we talked about coronaviruses are, what is a virus? This might seem very silly for a lot of you here because some of your backgrounds are expertise in viruses. But what I have learned from giving a lot of talks in India is that a lot of clinical doctors also prescribe antibiotics for viruses, viral infections as well. So one of the first things we go through is to figure out what a virus is. When we look at the origin of life, we have a common ancestor from the primordial soup that was there about a few billion years ago, a couple of billion years ago. We have bacteria, which are single-celled organisms that can be beneficial, that can survive on their own, that are independent and can cause infections. We have archaea, which are somewhat like bacteria, but use some other independent strategies for their own survival. Eukarya is you and me and everything from the yeast that makes your beer to plants and animals, trees and monkeys and dogs. When we talk about what life is, viruses are actually their own little group. They are the official definition of what a virus is. They are an intracellular parasite. Basically, it is considered alive inside the cell, and it's considered dead outside the body. And a virus is very simple. It is a very efficiently packaged self-replicating mechanism that contains the proteins that it needs for its own replication and the nucleic acid which carries its genetic information. Very simply, these are viruses. And the other important aspect to remember is in the central dogma of life, that is, when I talk about life, I'm not talking about viruses here, general life, we have DNA which encodes our genetic information, and the DNA is the chosen molecule for this because it is an extremely stable molecule that can be copied with a lot of reliability. And this is important to make sure our cells continue to function the way they're expected to. However, it is an extremely large molecule, and it's not efficient to open and read the DNA every time. So there are parts of the DNA which are copied into RNA, which is a relatively stable molecule. It can be read by the cellular machinery to make the proteins that we need in the body. What is interesting about viruses is not all viruses have DNA as their genetic information. Viruses have co-evolved with life to survive, to do whatever it takes to survive. Viruses can have DNA as their genetic material, double-stranded DNA, as is present in our cells. They can have single-stranded DNA. They can have a double-stranded RNA. They can have an RNA which has a positive sense. I'm not going to go into details of what a positive or a negative sense is. Let's look at it as reading it from right to left, and then RNA with a negative sense is something that we read from first, positive is when we read from left to right, and negative is when we read from right to left. We also have a positive sense RNA that can have a DNA phase and exploit that mechanism to make copies of itself. And then we can have ambivalent DNA, which can be some portions of it may be positive, some portions of it may be negative. These are all evolutionary strategies developed by viruses in order to exploit the host that it can infect. And then once it enters the cell, it forms the mRNA, which the body reads as its own and makes proteins. The virus then takes over the body, to the cells in the body, to assemble more copies of itself while destroying the host cells. Now, when we look at viruses, there are lots of ways of virus classification, and this is continuously evolving as we understand more and more about viruses. The broadest form of classification is we have DNA viruses and we have RNA viruses. In the RNA viruses, we have positive sense RNA virus, which meaning the genetic material is read from the left to right. And if the virus is enveloped, that is, if it has a lipid envelope around it, it's called an envelope positive stranded RNA virus. Coronaviruses belong to this category, meaning their genetic information has a single-stranded positive sense RNA, and their viral protein is covered by a lipid structure. And it belongs to the order Nidoviralis in the family Coronaviridae, in the subfamily Coronavirinae. There are 4 different groups. We have alpha, beta, gamma, and delta coronaviruses. The most important for us are the alpha and the beta coronaviruses. These have historically been known to infect livestock and humans. So obviously, it's of a lot more of commercial and it's a lot of interest to us in terms of life and commerce. We also have delta and gamma coronaviruses, which infect birds and aquatic life. When you look at what a coronavirus looks like, this is the different viruses that are in the Nidoviralis family. If you see, they all somewhat have a similar structure where they are enveloped, and this is what a coronavirus looks like. This on your right in the gray scale image is an electron micrograph of a coronavirus, and on the left is a schematic of a coronavirus virion. We have the inside, we have the RNA, single-stranded RNA, which is the genetic material, and here we have the viral membrane, viral envelope, from which covers the virus's structure, and it has different structural proteins to stabilize this virus envelope. But the protein of interest to us today is the spike protein. The spike protein is the protein that is initially seen by the body when an infection takes place, and the spike protein is what antibodies are targeted against, whether it's from a natural infection or whether it is from a vaccine. When we look at a coronavirus life cycle, there's a lot of information here, but I'll take you through this real quick because we need to understand all this to understand the origin of the virus itself. This blue color is your cell, and this green color is the endoplasmic reticulum. The dark green at the very bottom of the screen is the nucleus. The entire life cycle of the coronavirus takes place only in the cytoplasm. There is no function for the coronavirus to enter the nucleus of the cell. The virus binds a suitable receptor on the cell surface. For example, for SARS coronavirus 2, we have the ACE2 receptor, and this enters the cell. This entry takes place when the spike protein binds to the ACE2 receptor, and the virus envelope fuses to the cellular envelope, and uncoating takes place. At which point, the genetic material, the RNA, is released, and this RNA is processed by the cell to make more copies of the viral proteins. And then you have the viral RNA synthesis, where more copies of the genetic material are made from the proteins that are translated by the coronavirus genome itself. And then more copies of these proteins are made and the virus assembles, and then it's sent out to infect the neighboring cells. The main points, the bottlenecks that we can target in terms of reducing the seriousness of the viral infection, the first, of course, is preventing the attachment and virus neutralization, which is the strategy followed by giving vaccinations. The other points which I briefly touch are those antivirals that are given, which prohibits entry, which prohibits protein translation, and the other which prevent the viral RNA synthesis. And then there are also some antivirals that prevent virus maturation. However, these are a bit more tricky, because if we recall how the viruses exploit the cells, they take over a lot of the host proteins for their own replication. So we have to be extremely careful in designing antivirals, which can only affect the virus but not affect the host proteins itself, because we don't want to be poisoning the host. In some viruses, this does happen where we don't have a suitable vaccine, for example, HIV. But for coronaviruses, our best strategy is to prevent virus attachment and neutralize it in terms of using the vaccine. This is just a focus on what I just discussed. So we prefer the method where we prevent virus attachment and neutralization, where antibodies produced by the body are used to prevent virus entry. I'm just running through these. So this is what a cell looks like when it is completely infected by a coronavirus, by coronaviruses. This is actually a COVID-19 infection. The cell looks completely ravaged. A normal cell will look like this, where it is a lot of grayscale, and not like this, where it's completely ravaged and broken up, carrying viral particles. And these viral particles will either reinfect the same cell, which is inefficient, but that's just how things work in the virus world, but they can infect the neighboring cells and can be ejected from the body to cause further infection. Now, coronaviruses are zoonotic, meaning there are a variety of animal hosts for coronaviruses. I briefly mentioned there are 4 groups of coronaviruses: alpha, beta, gamma, and delta. And in this shortened list, we actually see coronaviruses can cause a variety of respiratory and enteric and other infections in humans and a variety of other animals. Interestingly, the interest in human coronaviruses came much later than it did in animal coronaviruses, because, for example, we have the TGEV, which transmissible gastroenteric coronaviruses, where it can wipe out an entire pig farm. So it's very important for the people that were raising livestock to prevent these infections. We have a canine coronavirus which can be lethal. We have feline coronavirus which can be lethal in kittens. We have a canine coronavirus for which there is a vaccine that has been in existence for many, many years now. So every time we look at coronaviruses, it appears as though there are coronaviruses everywhere that we look. And when we look at the timeline of coronavirus, the first coronavirus was IBV, infectious bronchitis virus. It was discovered in 1931. This was for chicken farms, where basically farmers were losing their entire livestock because this virus was spreading extremely fast in their entire farms. And then when we had the era of electron microscopy, where people basically threw in all samples under the electron microscope, we were able to identify multiple coronaviruses, the earliest being 229E and OC43. These are coronaviruses that cause a common cold. And over time, we have discovered how the virus actually exploits the host cell system for its own copies, and other coronaviruses. We have the emergence of SARS coronavirus, which came in 2002. I believe that was the time when the world sort of paid attention to what kind of damage coronaviruses can cause in the world. However, it was controlled pretty quickly because the virus was not spreading very efficiently among people, even though it was a droplet spread infection. Since then, there was NL63, which was discovered in the Netherlands. We have HKU1, which was discovered in Hong Kong. What is interesting Okay. What is interesting is most of these discoveries happen when people study the flu virus, because that's also a droplet transmitted disease, and it does annually cause a lot of death worldwide. And then with the emergence of MERS coronavirus, the Middle Eastern Respiratory Syndrome virus, we knew that coronaviruses could cause lethal infections, which had MERS coronavirus had a mortality rate of 30%, which is extremely high. However, the reason it was controlled was because it was again not a very efficient spreader. So now at this point, we knew coronaviruses were capable of spreading quite efficiently and were capable of causing death. And then we had initial data about the emergence of coronavirus, new coronavirus at the end of 2019, and then we started having the coronavirus pandemic in 2020. When we look at the coronavirus genome, it is a large RNA genome. This is a schematic of what the viral genome looks like if you just stretch it out and lay it down and put references to what each of these portions of the RNA genome does. So when we look at, it's 30 kb RNA genome. In fact, for an RNA genome, it is quite a long genome. Generally for HIV, it's about 9 to 11 kb, which is much smaller. It's only a third of the size of the coronavirus genome. The first 1/3 of the coronavirus genome, which is not represented to scale here, actually is important for virus replication, proofreading, and this proofreading activity is extremely crucial for coronaviruses, because if it does not have a proofreading activity when it makes more copies of itself, it is going to make many mistakes in this 30 kb genome, at which point it could easily render itself inactive, and it may not continue infecting anymore. And then we also have a lot of non-structural proteins that affect its ability to infect the host, stabilize the replication structure, and all that. Now, when we look at more parts of the genome, these proteins from this portion onwards are the structural proteins that are important for forming the viral structure itself. The reason I'm talking to you about these proteins is that these proteins actually help us identify the origin of coronaviruses, which is quite an interesting data in bioinformatics. So we have to look at error in replication. All evolution takes place by changes in our genome. We have evolved from a single cell bacteria, a functioning organism, simply by the process of evolution, which happens when changes in the genome takes place. The human genome has about 3 times 10 to the 9 base pairs. That is the DNA genome. And we talked about DNA being an extremely stable molecule. So as per this error-rated replication, every time our cell makes a copy of its cell divides, our genome will have about 3 base pair changes. If these base pair changes affect something extremely crucial, then that cell will die, or we'll have something like cancer. In HIV, which is a very efficient small virus, it has there is an error-rated replication of 1 in 70 base pairs. So if you have about 9,000 base pairs, you have about 1,000 base pair changes in every time the virus replicates, which is one of the reasons it's very difficult for us to form a vaccine against it, because the virus simply will find a way to escape it and go away. Coronaviruses are somewhere in between, which has 1 base pair change in about every 10 to the 5th, 1 base pair change in every 10 to the 5th base pairs, which is a pretty stable genome. However, this still does cause changes in the viral genome. So when we have these different changes that are happening, this is the schematic of the viral genome. And these different shapes represent changes in the viral genome. The virus will simply adapt to the changes that allow it to spread more efficiently. It doesn't necessarily mean that the virus will be more lethal. It doesn't necessarily mean the virus will cause more deaths or anything like that. It simply means the virus will evolve in the direction that allows it to replicate and spread more efficiently. As time progresses, we'll have the population of the virus which can spread the fastest to be the highest, and the population that doesn't really allow it to spread fast will be the lowest. So we have a lot of, so this is another thing that we have to keep in mind. And when you step back a little bit to understand about coronavirus, 30% of all common colds are caused by coronaviruses. We have, And like I said, coronaviruses are zoonotic, meaning anytime there is a close interaction between an animal and a human in large quantities. For example, we have these farms in places like China. Now, this is not a target towards China. Any place where there is a close interaction between humans and animals, frequently there is a chance of a zoonotic virus switching from its natural host to its intermediate host and then infect humans. So we suspect the origin of coronavirus, SARS coronavirus 2, could be bats. There is a lot of work that is done in terms of viral sample collection world over to help us understand the origins of different viruses. Viruses collected especially from bat tropics, and for some reason bats are especially good carriers of different viruses without getting infected themselves. These viruses found in China and Asia are somewhat similar to each other, even though there is probably no transmission between the the bat population that is there in the middle of China to somewhere in India. In fact, there are some caves near the Wuhan region where it's considered a hotspot for viral evolution, which is possibly one of the places from which viruses from the bats were transmitted to the local bat population, where the droppings infected the humans that were working in close proximity. Now, if you look at coronaviruses, they are all around us. Every one of these species, there is a coronavirus that is infecting it. So it's not quite a surprise to expect a zoonotic transmission from animals to humans. When we look at the ancient origin of coronaviruses, a very elegant study was based out of China where they used bioinformatics to understand how these viruses came in. So when we talked about the different viral proteins, we talked about and their rate of replication, you can backtrace the rate of replication to an ancestor of common origin. And it was realized that anywhere about 190 to 489 million years ago, coronaviruses probably originated, because the hosts for coronaviruses originated at this time as well. So the ancient coronaviruses are not a modern group of viruses, and they are probably one of the oldest group of viruses among us, just like how the herpes virus has been one of the oldest group of viruses that we live among. So bioinformatics was able to help us determine the ancient origin of coronaviruses. Now, briefly, I'll be talking about a phylogenetic tree to now understand how coronavirus, the COVID-19 virus and variants are evolving. When we look at what this typically is one of the ways that we look at a phylogenetic tree. This is the ancestral origin, and then you have various life forms that split in different branches. So the latest would be on the current variants would be on the rightmost end of this phylogenetic tree, and as you go backwards you will be looking at the ancestral lineage. These lines are not arbitrary lengths, they actually represent how many genetic changes are there from the previous branch. So just if you look at it to understand, and the another way of looking at it are these branches, where from the common ancestral origin you look at the different branches, and at each point you have another branching out based on the number of mutations that are there in the ancestral virus.
वो मुझे दे दो, उसको मैंने कुछ होमवर्क.
Now, I'm only going to talk about the current variants of interest in coronavirus, in COVID-19, because if you go to the WHO website, they talk about the different variants that are available. Now, we can look at all the different variants that are available, but that is also kind of pointless because not all of them are significant. How these variants are identified is worldwide samples are collected by different entities that sequence them. And when they do the sequencing, they look at the different mutations that have taken place. Now, most of these variants actually look at the genetic changes that have taken place in the spike protein. This is very crucial because our primary methodology to fight to prevent COVID-19 infections is vaccination. And the target for the vaccination is the spike protein. We have to ensure that the vaccines that we are currently administering are effective against these upcoming variants as well. Now, this is not so significant when we look at a whole virion vaccination, for example, like Covaxin, which is available in India. But when we look at the RNA-based, mRNA-based vaccines like Pfizer, they look at specific they use specific portions of the spike protein to generate immunity. It is quite effective. However, the problem is it cannot be applied even to a suboptimal level when the virus changes its receptor binding domain in order to escape the antibody effects of the antibodies. So right now, the virus, the variant of interest and the variant of concern, this was the Delta, and right now we have the Omicron. These different numbers, B1.1, 1.5, 29, or all these are simply scientific nomenclature given for tracking. However, for the common man's use, it is still considered the Delta or Omicron, the given Greek names because it is easy for people to remember and talk about it. So at this point, I will be taking you briefly to these beautiful websites where this website is called nextstrain.org. Okay, so these are the different variants of the Omicron itself, which have been identified, and these sequences have been deposited in this database from different portions of the world. So we have this 2A, we have 2B, and then we have a 20A, 20B, and all these, and these are the current variants. You can go and analyze each of these variants and what their changes are, how many nucleotide changes are there. I'm sorry, moving this cursor is disturbing to show what's there. If we look at this particular, let's say we look at this. This was identified, this is a clade 21K, and it was isolated on April 2nd, 2022. And there is a 64 amino acid change from the previously identified clade. So this data gives us how this virus is mutating towards. Now, this is not to say that we can predict the direction in which the virus will mutate towards, because we simply cannot. The viruses can evolve any number of methods to escape our surveillance and immune system. Okay, not this. Then we have the next initiative is called the GISAID, which is the global initiative for archiving influenza data, which I almost mentioned this before earlier, because one of the most common infections that we had worldwide was influenza, and influenza tracking worldwide has helped us figure out how to plan our vaccine strategy for the upcoming year. Now, the same system has been exploited to study the COVID-19 variants. So, for example, if we look here, we are able to track the different COVID-19 variants based on the sequencing data that is available. If we look at the map of the various tracked occurrences, we are now going all the way from the different. So if we look at each of these, we see the different variants that are available. So there is a constant, like for example. So from India, we have submitted 60,462 sequences, and it is seen what these sequences are taking us towards. So we are somewhat aware of what is happening around us in terms of our response time, which helps us react quickly enough to stop or control the spread of these variants. So, for example, here you can see in the UK it seems to only be Omicron, or in India it seems to be 97.5%. In a lot of countries currently the spread is, well, Austria, I'm not sure, because I'm not sure why it's 22.3, but it's again very important data to know, because perhaps a vaccine that is not working in other countries may be useful in Austria, and in countries like Ireland and Norway, it completely seems to be Omicron. So what is the future in which we will take this virus variants will move towards is something we don't quite know. And this is so important to track, because when we have an efficient antibody produced either by a previous vaccination or by previous exposure to the virus is fully neutralized and the infection is controlled. But however, when we have a suboptimal neutralization, we have viruses that escape the immunity, which also allows it to evolve further to becoming which also allows it to further escape the immune system, and it can infect more and more people. The problem with COVID-19, not just any COVID-19, but any infection, is not just the number of mutations that are there, it's how fast it can go from one person to another. And the faster it goes from one person to another, the more chances it has for developing more and more mutations, which will render our current immunization strategy, which can render our current immunization strategy to be useless. So how do we deal with COVID-19 in the future? We have to have complete equitable vaccine access everywhere. I know countries where people are getting their third booster shots, which is quite ludicrous to me, because there are people in different countries where they're not able to get their first vaccine either. So what's going to happen is, in the countries that do not have the vaccination available at all, they will become hotspots for evolution of more lethal, more efficiently spreading, possibly lethal variants. We also need to diligently monitor the samples to identify new variants at the earliest. Based on the information that I've shown you, this work is actually going on quite efficiently in a lot of countries in the world. We can perform some analysis of these mutations on current methods of prevention and treatment methods, which will help us perhaps stay at least on par with the virus in terms of preventing and reducing the COVID-19 infection. So with this, I end my talk, and I am now open to questions. I hope I didn't just bullet train my way through the talk with my speed, but I'm happy to take your questions now.
Good presentation, and I'm sure that people would have a lot of questions. So I open this forum. We have Dr. Chong also with us, our chair. Welcome, Dr. Chong, and a beautiful background. Okay, so can we have question answers?
Yeah,
Can I start, Surat? Can I start with a question?
Please, please.
Sure.
Thank you, Dr. Pavitra. Thank you. Is that correct?
Yes, absolutely, Dr. Chong. Thank you.
Okay. Hey, you know, I've been looking at some of these. I mean, there's monkeypox, there's severe hepatitis in children, and all the variants coming our way. Is there I understand that there's a connection with global warming as well, because as the planet warms, there's more jumping of the intraspecies jumping. Is that a truth?
There is preliminary data to indicate that as our environment changes, the viruses also adapt for their own replication. It's not a malicious intent. The virus simply wants to make copies of itself. There is some preliminary data to suggest that yes, the climate change does allow more novel, newer infections to come into play. One of the biggest fears is we have the permafrost region in different parts of the world. When the permafrost melts, we actually don't know what bacteria or viruses are in those regions. Well, a talk I'd given a little bit earlier, a couple of weeks back, talked about when we drilled ice cores from Antarctica, going as far back as a couple of million years ago, and samples were taken from these for analysis. Novel viruses were found in these samples. Whether these viruses are capable of infecting humans or not, we just have to wait and see. But there are viruses even in the permafrost from 2 million years ago, 3 million years ago. So let's see what the future holds for us.
I suppose this intraspecies jumping is also, like you said, due to the fact that humans and animals are living on top of each other. For example, in certain parts of rural Asia, and also technological change. Goods and people are moving across the globe, and of course you have demographic changes, population explosion in cities, blah blah blah. So it's all a big picture, and I always ask myself the fact that the human body is 37.2 degrees, and I'm sure most mammals are as well. So it seems that there's a predilection for viruses to head towards the 37.2 degrees. Is that right? So global warming is also in that direction, isn't it?
I don't know. That's a very interesting way of looking at it. Are viruses liking 37.2, evolving towards 37.2, or are we only interested in the viruses that survive at 37.2? We also have viruses that can infect fish. Coronaviruses, the can infect fish. So perhaps those coronaviruses have evolved to not particularly prefer the 37.2 degrees centigrade. And this is not the only time we have had. I know this is from the time COVID came up, people were like, oh, this is China, we should ban Chinese. I think that's a very ludicrous prospect because we had Nipah virus in India. And the whole xenophobic comment was the Chinese people eat bats. I'm like, that's not quite true. We had Nipah virus in India and we don't eat bats. So what do you have to say for that? It got me into a lot of trouble at that point, but eventually the people that wanted to understand what I was trying to say did understand. That these are simply accidents. We have these large pockets of hotspots of evolution where there is simply a lot of life, not just human life. We have animal life, bacterial life, viral life as well. I don't know why viral life is the correct statement, but yeah. So there is this. We also had plague in the 13th century, which came because we were trading. So this transmission, we've only become very efficient at our own movement, that the viruses have also adapted themselves to move along with us.
Thank you. One more last thing. I have a patient in hospital right now, or rather is my cardiologist colleague's patient. And he's, of course, has lymphoma, and his immunity, they vaccinated him, but his COVID antibody was 0. So he caught chronic COVID. He's now in 8 weeks. He's got 8 weeks of COVID. PCR continues to be positive with a low CT value. So it's a kind of chronic COVID. And I understand, for example, in certain parts of the world where HIV is not well controlled and so on, you have a tendency to get chronic COVID. Is this one of the biggest originators of the variants?
It could be. What is quite interesting is variants come up when there is not a lot of challenges to it, or there is some amount of challenges to it. So when there are suboptimal levels of challenge, the virus finds a way to sneak out. It is hypothesized that the Delta variant, which is one of the most interesting variants that escaped the immune system, probably came from a person who had chronic COVID and whose body had suboptimal immune response to it. So yeah, so it is quite possible.
So that's a scary part when you, let's say you have zones where you have a lot of these people with chronic COVID. My thinking is Omicron came likely from one patient. I hear views about that, right? And probably BA2 came 8 weeks later, just 8 weeks after Omicron,
yeah.
And 8 weeks later, so maybe another patient. It's quite scary because when we saw this chronic COVID patient in our ICU here, we were like a little bit astounded, and I'm sure the virus is mutating every day in his body.
Yes, that is scary. That is extremely scary. The most efficient viruses are not the ones that kill you in 3 days. The most efficient viruses are the ones that survive in your body, don't really do much, let you live a long, happy life while it changes its form and continues spreading. So that's exactly what you've summed up here.
Thank you. You really helped me understand a bit more about the variants and so on. Thank you very much.
Thank you so much. Dr. Bukhari had a question. Right. Yes.
Thank you very much. It was very nice lecture. I'm sorry I was late. I have another question related to the long COVID, because the monkeypox is now appearing in non-endemic areas other than the Central Africa or the West Africa. Is there any correlation? About 200 cases up till now, and more than 32 cases in the UK. Is there any relation of this long COVID and this monkeypox virus? Is there any mutation, or this is the transfer of the same virus coming from South Africa?
It's a very interesting question, Dr. Bukhari. I think a lot of people want to know. Everybody's overwhelmed with the kind of information that we have about viruses. a virologist like me always keeps track of the different viruses that keep coming up. WHO has a nice alert where you can ask for an alert every time a new virus is identified or tracked or whatever. But right now, this virus-related information is becoming headlines. So it leads to a lot of confusion among the general public. However, monkeypox is a completely different family of viruses.
I understand.
That is entirely unrelated to COVID-19. As for whether the people that have long COVID are more susceptible to monkeypox or not, I don't have the answer to that question. Monkeypox simply seems to have originated, and we are all very hyper vigilant about virology, virus-related information, which makes our mind connect dots perhaps where there were no dots previously.
Thank you.
Dr. Anjali, you have a question.
Thank you so much, Pavitra, for a very nice presentation. I just would like to know, there's a huge thing out in the media, again, regarding vaccines and myocarditis. So we all know that myocarditis is actually a bigger side effect from getting COVID-19 than from vaccines. But the prevalence of the myocarditis from getting vaccinated, is that more confined to young males, as we have seen in the studies, or is it something that is more widespread and not only related that much to males, but to females as well and older population after they have getting the vaccine? Thanks.
Dr. Angelique, I do not have the data to answer your question with a high degree of confidence. However, the one thing, and I am not a clinician, so I do not know the patient response to the vaccines or for COVID-19. Based on my understanding, like your first sentence was about the myocarditis is a much bigger concern from COVID-19 itself. I would still stick to the same thing, saying, as far as I know, based on the local information that I get from the clinicians around me, there does not seem to be a higher risk for females, younger or older, to develop myocarditis after any of the COVID-19 vaccines. There's a question in the chat box. In Pakistan, we had 2 doses of vaccine a month apart, then a booster dose after 6 months. Do we need a booster dose now that 6 months have passed? Typically, booster doses are given once a year. Whether at this point, I'm not sure who are the interested parties listening to this. At this point, we're almost vaccinating out of panic rather than a scientific analysis of when and how we need the vaccination. We're hoping to just jab people frequently enough, hoping that they will not get COVID again. In some ways, it is a good thing if the person has some lower immune response or a low functioning immune system. In other cases, that vaccine is perhaps more beneficial to someone who's completely unvaccinated. Dr. Monica, Dr. Basudev, you have a question.
Yeah, so I'm going to just make an amendment to your statement on the vaccination. So the booster recommendations in the United States is that if you're 50 and older, or you have an immune compromised state, the vaccination is meant for those, second vaccination is meant for those who are 50 and older and 5 months out from their first booster. Do I recommend a second booster? I definitely recommend a second booster. In those, looking at your scenario, look at your own personal health, look at the health of those who are around you, and then look at what the data is in terms of how many cases you have around you. What are the benefits of vaccination? One is whether or not you have access to medications to reduce morbidity and mortality, such as Paxlovid or Molnupiravir. The second would be also to very low chance, but it is found that maybe in approximately 15% that there's a reduction in long COVID symptoms. We have seen that reinfection is common. We are now documenting multiple cases of patients who are getting reinfected. They might have been infected with Alpha Delta. They've been vaccinated with their initial series, might have even gotten their initial booster, and now they're getting Omicron. So that's going to actually lead me to my next question for you, Dr. Venkatagopalan. Your presentation was fantastic. And to have your presentation now is very nice, to review the pathophysiology, the physiology of the coronavirus when we're so far into the pandemic. What you did was a great summary for all of us to remember again the basic biology and to understand the science behind the clinical approach that we have. The question I have for you is looking at the end game when we go from a pandemic to an endemic situation. We have seen multiple variations and we're seeing the antigenic drift.
Yes.
We went from alpha, beta, delta, gamma. We then developed Omicron, which was kind of on a different arm. We went from BA1 to 1.1, then we went to BA2. We now have BA 2.12.1, that is the major form in the US. And now we have a new BA 4.5, which has more immune escape than the prior Omicron. So in my understanding of how these antigenic drifts happen, in terms of how long we are expecting this pandemic to occur, these new variations seem alarming to me. Like, I don't feel like we are in the tail of our pandemic. I just feel like the minute we turn around, we now have a new variant that we're concerned about. So knowing from your personal experience and your studies of the coronavirus, how can you take what you learned from 10 years ago during your PhD training to the pandemic that we are now? What do you make in terms of your understanding of how is this pandemic going to evolve from the position that we're in? We have multiple states of natural immunity where people have been infected with whichever variant. We now have partial, vaccinated from multiple different types of vaccines. And we do know that there's altered T cell responses, neutralizing effects of the different vaccines that are out there. But we're also seeing that these vaccines are not very long lived and that there's now recommended multiple doses of these vaccines. We're still waiting for the second generation of these vaccines. So we're in this situation now where everyone has COVID fatigue. Where are we going to, or how do we look for the end? What is it about this pandemic that will make us feel that, yes, we are finally in the home stretch? I don't feel like we're in it right now. I know I gave you a lot of information there right now, I'm sorry, but how is this pandemic going to end, I think is what I'm asking.
That's fine, Dr. Vasudev. The reason I went over the basic history of the basic life cycle of coronavirus is where I come from. A lot of the clinicians I deal with still believe ivermectin works. No offense to all the doctors here, but this is something that we really deal with on a day-to-day basis. So so I just wanted to put that information out there that this is the direction we need to be going into. So a couple of things that we need to look at. We are still very early in terms of understanding how COVID-19 works. 2 years, a little over 2 years in a pandemic of this scale is unprecedented. The earlier we just all died. This is one of the situations where we are doing the best that we can do. The next stage of response in terms of what the end game would be, I don't know. We probably, though no swabs are here to stay in our lives, they are not going to go anywhere. So even when we have the flu vaccine, which predictably goes through an antigenic shift and drift, we have gotten relatively good at predicting what the vaccine could be that is efficient across the world, and we are doing it. And this is after many years of collecting data. At this point, we are still very much in the preliminary stages of understanding which direction the virus might go in. And every time we seem to be putting one roadblock, it escapes and gets out the other way. And the problem here is not just developing the second generation vaccine. The problem is also vaccine availability to everybody. You give someone 5 doses of the vaccine, boosters and all of this, you have a population that is completely unvaccinated. So unless we are able to reach some level of equality among people with their access to vaccines, and of course we have the craze is that still don't believe, that there is a significant number of people that don't believe in any of these prevention methods. They want a natural immunity from the infection. Let's forget the fact that, you could actually die from the infection. Or we have people that still do not want to mask up. We have a lot of brickbats coming our way as well in our process of trying to prevent this infection. So as we go, we probably, like I mentioned in this slide, it'll probably be continuous monitoring, developing newer vaccines, and having equal access to the vaccines in the future. How long this will take, when this will happen, I cannot predict. And in the meantime, we just do what we can with what we have. So that is a long, roundabout answer to, well, I don't know what we need to do.
So I hear from you that we're not close. We're not close to the.
No, we're not nearly close. Yeah. We are not nearly close. Thank you, Monica. If I may move to Dr. Jamal Uddin from Bangladesh, that'd be great.
Thank you so much. Really, my question is actually, you have just raised a point, very important point. That's you have said that terminology we initially used much. This is novel coronavirus. So, but you are saying that it's from 1931 there was coronavirus. So I think that was not a good actually account. And my second point is which the factor actually causes this more virulence actually in terms of infecting the human being seriously. And initially we have seen in Italy and others when we are not having vaccines, a lot of actually casualties were there. After that, we just found out that yes, after postmortem in China and Italy, that yes, it's creating coagulation and intravascular coagulation, and so that people are dying like this. So we went for the anticoagulation and other measures. Okay. So my question is, what is the actual factor? What's the factor that after such a long time we have seen that after March, you say it was contained much. It was not too much contagious. Okay. In terms of RO, it was not so high, but the mortality is more than 30%. What you were saying, because of March. But what about this COVID-19? And what is the factor actually mostly? It becomes more virulent and more serious for the human being, so that we can just understand about this.
So this COVID-19, when it first came out, was called novel coronavirus. This particular virus is a novel coronavirus. As a family, coronaviruses are well studied. So the initial name for this coronavirus was simply novel coronavirus, before the virology taxonomy group decided to call it SARS coronavirus 2. That's the simple answer to the first question. How and why this MERS was 30%, had a 30% mortality rate versus COVID-19, which is, let's not go into the number of deaths caused by COVID-19 because it seems to be a highly charged political issue, at least in our country. What happens is any virus, you look at it, it's not the virus that is the most lethal that is very effective, or even bacteria for that matter. The viruses or bacteria that allow you to live a long life and spread the infection for a long time is the most successful virus or bacteria. We have tuberculosis, which has been around from Egyptian times. We have polio that have been around from Egyptian times. And we have HIV, which is a very small virus, like 11 kb, that's all there is. It's so tiny that you would expect that our immune system would take care of it. But it has exploited our body in such a way that it escapes the immune system but does not horrifically kill you. Which means the person infected might not know they are infected. A person infected might not know they're carrying, and they will continue to spread the infection. So the evolution of this novel coronavirus, SARS coronavirus 2, probably happened when one person was infected by a sample somehow from a bat, and it evolved in this person's body to be a virus that spread very well and initially was causing a lot of death. And causing a lot of death is not an efficient way for the virus to make more copies of itself. So it has simply evolved itself in a way where it can survive and infect. The death is a side effect for the virus that is immaterial to it. It just wants to survive as much as possible for as long as possible, in fact, as many people as possible. Moreover, and we were initially, we have been extremely good at developing at vaccines. So as time progressed, the virus may not always evolve into this non-lethal format. It will simply evolve better to survive. It could become more lethal. It could become less lethal. So there's no way to predict which way it will go. But we also have added and mixed the vaccine or previous infections that help this, that make this virus to not become extremely lethal. So these are the two ways in which the direction in which the virus is evolving. And I hope that somewhat clarified the question. MERS was easy to identify because these people had wide coughs. So the moment you knew you had it, you were isolated. COVID-19, you don't even know what you have. So at least for 3 or 4 days, you're going around spreading the infection.
But the SARS also was having the same symptoms?
SARS coronavirus, which came in 2002, it did cause high fever and severe cough, but it was not as good at spreading as SARS-CoV-2 is. See, every time an infection takes place, one viral particle is not going to infect you. You need a minimum cluster of viruses to infect you. And if the virus is better at infecting with fewer number of viral particles, then it gets better. That's one of the reasons COVID-19 virus, the SARS-CoV-2, is also good at spreading so easily, because the number of particles, in fact, the number of particles that was needed to infect from the alpha lineage to the Omicron lineage has come down by, I think, about 20 odd times. Thank you, Dr. Jamal. May I have answer your question, Dr. Kaisa Sajjad?
Thank you. Another thing is just about the monkeypox. Can I just ask you one thing? That whether we are worried too much, because I have seen it is a history says that in 2017 in Nigeria there were 500 suspected cases. Out of that, only 200 were confirmed, and in US only 70 cases were in there somewhere in 2003 or 4 like that, or 2012 like that. So it is contained much. So why we are so much worried about it? Because of these mutations only, we are just worrying too much throughout the world. It's like this. New mutations expecting?
No, we don't expect a monkeypox outbreak at any point, but it is known to happen. And a lot of the other thing is, most of the world is not vaccinated against any sort of pox right now. We are not vaccinated. If you're born after 1973, 74, I don't think you're vaccinated against the pox family of viruses. So there is that fear as well. But this virus does not spread very efficiently among people. And because people have visual symptoms of infections, it's easier to isolate them. So yeah.
Okay,
thank you. Dr. Kaisa Sajjad, may I answer your question? Doctor, if you could please.
Can I answer this question?
Okay, thank you. Now is it okay?
Yeah.
Thank you very much, Pavitra, for the excellent presentation. I just want to share what you have mentioned about Pakistan. The vaccine which is being given to the people, basic vaccine was Chinese vaccine, and which was Sinopharm and Sinovac. And then after 6 months, they have booster dose, Pfizer booster dose. Most of the people have a single booster dose, but most of the doctors, paramedics, you can say the healthcare workers have 2 booster doses after a month gap. So this is an information, but I would like to know, is it a good things to do to have a booster, a 2 booster for a healthcare workers or everybody of a Pfizer, or what you suggest that after 1 year or 6 months, they have to have another booster, or every year or every 6 months? This is my question. Thank you.
Dr. Sajjad, I think I will redirect you to Dr. Monica Vasudev's comment about look around you to see what your scenarios, your exposure levels, your health status, the contacts that are around you. So that would be the logical way of going towards getting a booster or not. And mixing and matching vaccines in terms of the primary and the booster has been shown to produce a much more effective immune response than having the same booster given to you over time. One of the questions here in the chat box, Dr. Angelique had talked about natural immunity versus waning antibody from vaccines. If I may answer that question in a moment, coronaviruses actually have a way to suppress natural immunity. When you look at the viral protein, the RNA is bound by this protein called a nucleocapsid protein. This protein actually prevents the cell from signaling to its neighbors that it's infected. So the immune response generated from a natural infection can be reduced by this protein as well. So this is one of the reasons why I always say a vaccine is better, because you have nothing suppressing the immune system from actively responding. So we've had patients where I have tested samples where they have COVID-19 14 days and then 2 weeks later antibody response nil. You have nil antibody response after a COVID-19 infection. And this person does have a normal, full functioning immune system. It is not a person that has a low functioning immune response. So, my theory on this would be that having a natural infection does not automatically give you the kind of protection a vaccine can give you, because there is not fighting forces acting in your body during the time of infection. Dr. Sajjad, do you have any other questions? Dr. Chong, please go ahead.
Dr. Pavitra, can you just rehash what you just said, that last bit about
the nucleocapsid protein?
Yes, just rehash that a bit. I didn't quite understand it.
Okay, I'm going to go back to my presentation for a moment, and I will take you through this. I didn't talk about it because we weren't really talking about it. Yes. So when we look at the coronavirus genome, there are known proteins that suppress different arms of the immune response. For example, this protein, there are a lot of proteins that are here that are important for the viral replication process, but there are some which are known to suppress cellular immunity. The cellular immunity, in terms of how the cell signals to its neighbor, the interferon response is all brought down, can all be brought down by some of the proteins over here, some of the non-structural proteins over here, and this nucleocapsid protein over here. So you might have an active COVID-19 infection, but your immune response might not be as efficiently as you would like it, because the virus is actively fighting against you developing an immunity against the virus. This is one of the reasons the quality of antibodies that are formed after any coronavirus infection. I am not talking about COVID-19 here. I am talking about OC43 and 229E. The quality of antibodies that are formed, the quality of memory cells that are formed are lower, simply because the virus is actively suppressing this immune response. And this is the reason why we get common colds year after year. You would have had a coronavirus caused cold this year and then the next year too. The reason being your quality of immune response is poor from a natural infection, can be poor from a natural infection.
Thank you very much.
Thank you. Any more questions? All right. I hope that's okay.
Questions on the floor?
Excuse me, can I just ask?
Yes, Dr.
Can I
Actually,
just ask?
go ahead.
So thank you. Sorry to chip in. Pavitra, is there any article that we can refer to regarding what you have said? Is there any place that we can just
there's a lot of papers. One actually came from my own lab. I don't remember the year. It was by my colleagues Yeye, and I think the corresponding author is Hope B. It was in JV or virology, probably 20, 15 years or so back. This should be it. It was a very interesting collaborative effort between our lab that was studying coronaviruses and a neighboring lab that was studying smallpox virus. So these guys used to work late at night, and then they were like, what if I put one of my proteins into your virus and see what happens? I can't say this in a non-scientific community because people will be like, oh my God, this is bioterrorism. It's not, this is what researchers do. You could try these terms to find the paper. If not, I will send the link of the paper to Dr. Sanchita, who will be able to share it with you on your forum. Okay.
If there are no more questions, shall we call a close to this meeting? Sarah?
Dr. Tong, would you like to just give a small update on what's going on with the Singapore dengue?
Oh, dengue. Well, my son got dengue.
he's okay now.
I tell you. So my whole family has gotten dengue, and we've hit 10,000 cases, and it was and we're in May only, just the start of the season. We haven't hit the peak of the season yet, and yeah, I've got some interesting story to ask Pavitra about this. So 10,000 cases, and yeah, so my whole family's gotten dengue.
So Dr. Chong, is there a casualty?
Yeah, I think there's some. This year, I think so far I don't recall any massive numbers, but in 2020, the number of dengue deaths was higher than COVID deaths in Singapore. So yeah, so last year was only 6,000 cases, but this year is already 10,000 and is only like 1 or 2 months into the season. So it's really exploding. But what was very interesting was many years ago, our Institute of Cell and Molecular Biology here, I remember a presentation by Edison Liu, who was just, who has since retired and gone back to the US. He actually had his institute collect all the blood from the GPs. Okay, what they did for the GPs was, if you have a suspected dengue case, you draw the blood, we'll send it to our lab, and we'll run the IgM, IgG, and the NS1 for you, free. Okay, so diagnosis was free. But in return, they were collecting the data on the flavivirus. Would that be RNA? I think RNA, right? So they were collecting so all the specimen from across the island in different spots. And he presented the data very interestingly at a meeting, and I still remember this. And he actually said this. He says there is something in the data that shows that the mosquito is transmitting the virus to the larvae. So he mentioned something like that to me, to the whole audience. But it's been many years, I've never heard anything more about that since. Any thoughts, Pavitra?
No, I'm so sorry. I was a little bit distracted, but I remember recalling this talk, this very interesting talk in virology, ASV, American Society of Virology. But yeah, I've not heard anything from this in follow-up either.
Yeah, so it's fascinating because I just wonder to myself, I still remember what he told us many years ago. I wonder whether the mosquito is actually transmitting the virus to the larvae. I mean, there'll be a game changer. Scary and yeah. I mean, and in Singapore, because we have the Aedes mosquito, we're so terrified when we introduced flights to Brazil. We were so terrified that the yellow fever will come, same vector. So this is something we really our people here lose sleep over. Yeah. So that's
what is that project actually? What about that project which Singapore took some sort of
Wolbachia, the Wolbachia project.
Yeah. What about that? Why is it working?
so far with the outbreak, we don't see any. We don't see any
Anyways,
results,
okay,
you know.
okay.
So
We're very interested about this actually.
Yeah.
Even I don't know what is the factor behind, because we don't in Bangladesh we have got this spread, okay, but it's not in that scale which we are seeing in Singapore. But
Yeah.
you have a very better healthcare program, and so why it is spreading there actually? What is the reason? You find out?
we in tropical schools is taught that Singapore has one of the best mosquito eradication programs in the world because we got rid of malaria. You know, but even then, it's a huge challenge in Singapore trying to control dengue, and the mosquito can just grow eggs in slivers of water. You know, just slivers of water. It's just amazing. I mean, you know, I don't know what sort of commando mosquitoes we got here. You know, so yeah, but if there's this Wolbachia is a bacteria that is found that comes from, I think originally the Australians are the ones who are into this, and we got a technology from them, and we infect the Wolbachia into the male mosquitoes. They mate with the female mosquitoes, and the Wolbachia stops the replication. I mean, the larvae and so on, or the egg form. You know, the reproductive process. It somehow enters the reproduction system of the female mosquito, and then it's supposed to work that way. But so far, with our 10,000 cases, I don't think we are. I don't think we're seeing much effect on that.
Yes,
You know, so it's a challenge. It's a challenge in Singapore right now, dengue. Yeah. Thank you.
Yeah. Yes, I think we have also seen some cases in Bangalore, in Delhi, in India, in last 15 days. So I just thought, you know, the cases are like, you know, it's the same thing which is happening over here.
Yeah.
Okay, so shall we end this? I think the speaker has left because she just texted that her son is not well. So I would like to thank her, you know, having this session with us. It was a great session and a great discussion. So if we do not have anything else, we will end this session. And next week we have an you know, we have a session on intervention radiologist for COVID-19, and we have a speaker who will talk on this topic, and I think it's a very relevant topic. So see you again next week, same time, same day. That will be 113th session. Thank you so much for everyone for joining in, and
Thank you.
take care. Bye.
Bye.
Thank you.
Bye.
Okay, Suraj,
Bye.
thank you very much.